Hi everyone. In this lab, we're going to talk about molds. So everyone has seen molds on various surfaces, foods such as bread. And when you see, so here is mold grown on a certain type of agar plate that we talk about. So this is obviously not microscopic because we can see it with our naked eye, but molds do have a microscopic portion of their life that we will talk about. And here is mold under the microscope. So here it is on an agar plate. And if you take a part of this and you look at it under the microscope, this is what it could typically look like. So we're going to start off with some background with molds. Molds are eukaryotes, so they're eukaryotic organisms. They're part of the domain Eukarya, and they're part of the kingdom fungi. They are multicellular, unlike bacteria that are unicellular. They cannot move, they're non-motile, and they're filamentous. So all mold begin as microscopic spores, but then they eventually grow and they become macroscopic. So that's when you can see it on an agar plate, on bread, mushrooms, for example, mildew. They're aerobic organisms. So if you ever have mold contamination, one way to get rid of them is to remove the oxygen. So they like moist, oxygenated environments, and they're heterotrophs or chemoorganoheterotrophs, meaning they need both an energy and carbon source from organic matter. So they can't make it themselves. So if you want to try to get rid of mold, I would take away their oxygen because they're aerobic, and I would take away any food source. And keep in mind that with mold, they grow so easily and so quickly. So anyone who's ever had mold contamination knows about this in their house. And these organisms also secrete exoenzymes. So we talked about exoenzymes with the metabolism lab. Exoenzymes are when organisms make enzymes and then the enzymes are released outside the cells. And then these exoenzymes help them digest food. And then when they break down the food, the smaller components, the monomers, can come back into the cell. So they decompose organic matter for energy, and most are free-living, most are harmless, but some can be pathogenic. Okay, now we're going to talk about the life cycle of molds. Molds can reproduce asexually or sexually, so they can do both life cycles, but most commonly is the asexual life cycle. So that's what we're going to talk about. And mold classification is based on their reproductions of sexual or asexual. So again, asexual is the most common life cycle in molds. So the way asexual reproduction happens, so the way a mold reproduces or makes more of itself, is first of all, you have spores that germinate when conditions are good. So maybe there's a lot of oxygen, a lot of nutrients, so they germinate. That's what we mean by conditions are good. And I want everyone to keep in mind that fungal spores are very different than bacterial spores. Bacterial spores are resistant to destruction, resistant to so many things. Fungal spores are not nearly as resistant as bacterial spores. So when conditions are good, they're favorable, you have nutrients, you have oxygen, they germinate. Then the spores will form single threads called hypha, and the hypha starts to grow, and then a lot of hypha, so here's a single thread, so here's your spore, then it starts forming hypha, and then you get more hypha, and we call that hyphae. And then when the hyphae interconnect, and there's a lot of them, you form mycelium, which is a fuzzy mat. This is typically what you see on your food. And then if conditions get bad, again, the mycelium will produce spore-producing bodies and release spores, and then the spores can be carried out to better environment through winds, insects, and animals. So again, this is the life cycle. So we get spores, they germinate into hypha, hypha become hyphae, so more threads. And then when you have a lot of them, they interconnect to form mycelium. And then when conditions are not good, again, you get spore producing bodies that produce spores. And so this is the asexual life cycle of a mold. And keep in mind that we identify mold. So if you have mold contamination in your house, what scientists typically do, if any of you guys have ever had this, is someone will come out to your home and they'll take a sample and they will identify the type of mold based on agar identification as well as microscopic morphology under the microscope, because some mold species are a lot worse than other mold species. So molds are very important. They have benefits and they're detrimental, just like most organisms. Their benefits are they're very important ecologically. So without molds, we would have dead bodies that don't decompose. They're important for the decomposition of organic matter, meaning the decomposition of microbes, of animals, of humans, and recycling nutrients back into the soil, so getting carbon and nitrogen back. They're also important for human use. So one of the first antibiotics discovered is penicillin. And penicillin comes from a mold called, that is Penicillium chrysogenum. So that's where we got our first antibiotic. And this story is really cool. The scientist who was working in lab had grown bacteria on an agar plate and left the agar plate on his bench. And he had left it there for a few days because the story, I think, is that he was sick. And he came back later and he saw mold on the agar plate, but where the mold was, there wasn't any bacteria growing. So he knew that the mold was killing bacteria. And it's also important for food production. So some cheeses are made by mold, such as blue cheese, soy sauce, a lot of different foods that we get. Now, mold can also be detrimental. They are really bad for contaminating certain labs. So if you work with bacteria in lab, when you have mold contamination in lab, it's really tough because it's very hard to get rid of them. So they're a source of contaminants in various research labs. They can cause food to spoil, as a lot of you guys have seen, and they can cause allergic reactions. So a lot of people are allergic to mold spores. A term I want everyone to know is mycoses. So they can cause diseases called mycoses. Mycoses are opportunistic fungal infections, meaning that they infect people who are immunocompromised. So immunocompromised could be someone who already has an underlying disease, or for example, a pregnant woman is immunocompromised. So again, mycoses are opportunistic fungal infections. They take opportunity of someone who is immunocompromised. And we have superficial mycoses. So superficial means that it is on the surface. So that's like ringworm, athlete's foot. Some people have fungus on their nails. It's very common on toenails because people will wear socks, and if their shoes get wet, you have this moist, possibly aerobic environment for mold to grow. And then systemic mycoses, systemic means when it spreads. So these can be potentially deadly. And we're going to talk about two various infections that can be deadly. Okay, so in this lab, had you been in lab, we wanted you to look at mold under the microscope. So typically mold, we grow it on Sabra Agar plates, and this agar is a selective medium. Remember, medium means whatever we grow the microbe on. So the medium we use for mold is Sabra Agar, and it's selective media for fungi because it has low pH, meaning it's acidic, and it has very high sugar concentration. And by having low pH and high sugar, you inhibit bacterial growth. So it's selective for just fungus to grow. And once you have it growing here, so let's say someone has mold contamination in their house, you can just get a sample, you grow it on Sabra Agar, and then if you want to look at it under the microscope, what you do is you take a piece of tape and you just stick some of the mold on it, and then you put a stain called lactophenol blue stain, and the lactophenol blue stain lets you see which life cycle, which part of the reproductive life cycle the mold is in. So we're going to look at slides and talk about it. But again, the take-home message from this slide is that we grow mold on Sabra Agar, which is a selective media that inhibits bacteria. It selects for fungus because it has low pH and high sugar. And then when we look at it under the microscope, we add lactophenol blue stain. So here is a nice image of microscope pictures looking at mold in various life cycles. So again, we have spores. When conditions are good, they germinate to form hypha. So here are spores forming hypha. Hypha are the threads. Here's a lot of hyphae. Anytime you see all these strings, they're the mold hypha. And then when you get a lot of them together, they form a fuzzy mat called mycelium. So here's a nice microscopic image of mycelium. And then when conditions are not good again, the mycelium will produce spore-producing bodies. So here are our spore-producing bodies. So all these images show spore-producing bodies. And again, the stain used in all of these is lactophenol cotton blue stain. And this is the asexual reproductive life cycle of mold. So just know that these here show hypha, this is mycelium, and then these are images of spore-producing bodies. Now we're going to talk about two diseases, two pathogenic molds. So there's a mold called Aspergillus, and it causes the disease Aspergillosis, which mainly occurs in immunocompromised people. Remember we said that a lot of mold causes mycoses, which are opportunistic fungal infections. So the cause of Aspergillosis is mold. That's the etiology. The symptoms, it can be asymptomatic, but typically the main symptom is it's hard to breathe. So you get this mold in you and it becomes very hard to breathe from the fungal spores, which you inhale and then they germinate inside your lungs, because lungs are aerobic and these are aerobic organisms. Now, in immunocompromised individuals, the disease can turn bad very quickly, just like we see actually with COVID-19. So COVID-19 is a virus. It's not related to this, but you guys have all heard how it's a lot more fatal for people who are immunocompromised. So that applies to everything. So with Aspergillosis, it can be a lot more fatal, again, for people who are immunocompromised. The disease can turn into pneumonia and spread to other parts of the body, which can become very bad. The way that you get Aspergillosis is by inhaling fungal spores, and the treatment is antifungal medications. Now, if it becomes really bad, you need surgery to remove it. And the prevention is, if you are immunocompromised, especially to avoid dusty environments where spores are found. So here are images of Aspergillus, the mold right here. So here's an image. Here's a nicer image that I'm covering, but you guys have the lab with you anyway. And then here's Aspergillosis. So it is the mold infecting the lungs. And again, the spores will germinate in the lungs because the lungs are so aerobic, and this is an aerobic organism. And here is an image of Aspergillomas, and these ones have to be surgically removed. Now, another mold that we're going to talk about is Coccidioides, and this causes the disease coccidioides, which is known as valley fever for most people. The reason why it's known as valley fever is because we see this mold and mold spores in northern Mexico, southwestern US, San Joaquin Valley of California, where we see this mold. So doctors who work in this area have a lot of patients come in who have symptoms of like tuberculosis, possible lung infections, but really most of the time they have coccidioides, this disease, and it's caused by this mold, and the symptoms are flu-like. Again, in immunocompromised people, just like Aspergillosis, this can spread throughout the body and it can start off as pneumonia, a really bad lung infection when you get lung inflammation, and then the mold can spread everywhere. It's also transmitted by inhaling fungal spores. So it's endemic, and meaning it's common, these mold in northern Mexico, southwestern US, and then the San Joaquin Valley of California. And it's the same thing. So if someone has it, you treat them with antifungal medication and possibly surgery. So here's what Coccidioides looks like, the mold. Here's what it looks like under the microscope. And here are endospores in lung tissue. But here again is the mycelium of the mold. And here are more images of the mold itself. So here is an image of someone inhaling the mold spores and it causing the disease. So the spores get inhaled, and then in lungs they germinate because your lungs are very aerobic. Here are pictures of Penicillium. So this is a mold. This is where we get our antibiotic penicillin from. And so this is what Penicillium looks like under the microscope. So here's images of Penicillium chrysogenes. This is a microscopic image, and all of these again were stained with lactophenol blue stain. And here is an agar plate. Here is the mold. This is bacteria. Around the mold we see no bacteria growing, which is great. This revolutionized medicine. And then here is the mold growing on Sabra agar plates. Here's Rhizopus. Rhizopus is a mold that typically causes bread mold. So when you see mold on your bread, this is the mold species that's causing it. This is what this organism looks like under the microscope. And what I want everyone to know is the domain and kingdom that molds belong to. So again, they're domain eukarya, they're kingdom fungi. We talked about them. Molds are non-motile and they're very aerobic organisms. They differ from bacterial endospores. They're not nearly as resistant. And then we talked about beneficial and detrimental aspects of molds. Go over that. Opportunistic infection is an infection that takes opportunity of someone who's immunocompromised. So opportunistic infections cause infections in individuals who are immunocompromised. Mycoses is an opportunistic fungal infection. And then know the details about Aspergillosis and Coccidioides. I can't say this. You guys, I think, got the point. And then the name of the mold that we get penicillin from. So we talked about Penicillium and showed the pictures. Know the asexual life cycle of a mold. So how the spores go to the hypha become hyphae, all the hyphae interconnected become mycelium, and then the mycelium, when conditions are not good, could go on to produce spore producing bodies. So know what they look like under the microscope. The medium that we grow molds on is Sabra Agar. Why is it selective? I talked about that. And then the stain that we talked about, so lactophenol blue. So go over all of these things for mold.